So again, once more, many cells, sorry, the same type of cells, but different diseases. So I don't know if one could say, like this guy, well, I mean, it's working, it's enough you're using. So these cells manage to do so many things. This is more or less is what I share with James' presentation. Not only that, in our experience, we have been showing, for example, in this publication where we have used placenta-derived mesenchymal cells in lung fibrosis, we have been showing actually that the engraftment of these cells that are in this arrow, I mean, this was in an allogenic setting or in a xenotransplantation, so human cells into mice, or here are two different strains of mice that we have used. I mean, this arrow indicates presence of cells at day 14, so we cannot even talk yet about engraftment, but there are few cells, I mean, to what the type of effect that we observe. So, many diseases, the same type of cells, even we have a few cells engraftment. Well, what's going on here? Is it really true that these cells are something magic, or are we just using because we have it? That's the position of James. My way of looking has been, do these diseases have something in common? Because that could explain that they act on a common mechanism. And I think, yes, they do, all these diseases that we have been mentioning having something in common. They have some inflammatory processes. They have some dysregulation of the immune system. And if you think carefully, all the degenerative diseases, different type of disease, they start from an immune dysregulation. So maybe, and then I will show, this is why I managed then in this way to link the two presentations I wanted to do, maybe the immunomodulatory properties, so that are at the base of the action of these cells, could be, I'm not saying they are the only mechanism, but are for sure one playing a big role. This is for me the only explanation I can give why a cell type can be working in Crohn's disease, can be working in spinal cord injury, can be working in liver fibrosis. Well, the only way, it's hard to imagine a cell that is able to differentiate toward everything, and we did not demonstrate this, neither we did, or many groups did not manage to demonstrate this differentiation, even though I'm aware of some. But what probably everybody managed to demonstrate are the immunomodulation that is going on there. So trying to summarize what I said so far is that no matter you may have an injury, you may have a disease, well, you have what's going on here. So you will have an acute inflammation as a starting. This acute inflammation, you know, the inflammation, you need it to resolve your problem. In fact, you will have recruitment of activation of inflammatory cells, you have these inflammatory cytokines, and what's going to happen? If you are lucky, when you have an acute inflammation, you're going to have a resolution of that. So, okay, you're fine. But you might have other situations where you have a progression and you have a chronic inflammation. Or you might have your body is trying to heal, but with involvement of fibroblast, myofibroblast, collagen deposition, and this will give us fibrosis. So different type of diseases, imagine that this is where you are going to start. And if you do not have a resolution, you are going to either go into the chronic inflammation or into fibrotic processes. So what I think the mesenchymal stromal cells do, and then we will see if there are differences between the different sources eventually, but where these cells probably play a role is somewhere in this either progression toward the acute or in helping this healing process. And I will tell you why. So as mechanisms of repair, I summarize what I'm going to show you, is that we will see these anti-inflammatory properties, so they are able to block T-cell proliferation, they decrease pro-inflammatory mediators, They increase anti-inflammatory mediators, and they induce a polarization toward, for example, M2 macrophages. We've been shown they have antifibrotic properties, so there is a reduction of these profibrotic factors. And they also bring trophic support. So yesterday there was a discussion about cells that could manage to have a healing process because they bring trophic growth factors. Well, these cells are able to bring actually all of this. And this again explains to me why they might play a role in different types of diseases. And so the mechanism they have is very likely is that once you, in an environment where you have injury, where you have all inflammation, you manage to repair, to take away this. Now you have your resident stem cells that can do the job. I think there is no matter that you can have the most potent cells, but then when you transplant it, you need to imagine that this is going to communicate with the other cells that is there, so it's very complex. But if you are in the process where you are at the right step of this degenerative process and you have only the inflammatory process ongoing, and if you block that, and if you are able to repair the tissue, you probably could have now an ongoing regeneration of the tissue per se. And I think that this is probably, at least in my opinion, a way to look at regenerative medicine. And I think it will bring more success than the way that we've been looking so far of doing cell replacement. So a cell that becomes something else. Because we did not manage. The only way, the only system where this is working is in the hematopoietic system. In the others one, I think we are having a lot of trouble. But maybe we can look in this way, regenerative medicine. So now let me tell you something about the amniotic membrane. So what I told you so far, it's valid in general for any type of mesenchymal stromal cells, no matter where you take it out, because you see it's a basic mechanism. So of course it's an interpretation, but I would say based on the type of factors that is released. I want to tell you something about the amniotic membrane because I think it's a good source of cells. And okay, there are many out there, but I think this source of cell can become interesting because, well, it's easy to procure. It doesn't hurt anybody when you get this one, because I'm talking about amniotic membrane from term placenta. So this is biological waste. It doesn't cost. So I think it has a lot of advantages. And let's see the properties that these cells do have. First of all, let me, in case somebody of you is interested, well, when I talk about the amniotic membrane, I'm talking about this protective membrane that is around here, the one that makes the amniotic sac, the so-called. How is the structure of this membrane? It's very simple, actually. There is an epithelium. And as we all know, there is no epithelium without a mesenchymal layer. So there is these epithelial cells, and then there is an area very rich in collagen, and there are inside this area some mesenchymal stromal cells. We have been characterizing these cells, and so this slide is actually showing how we can detach amniotic membrane very simply from the entire placenta, and we can start having, if we want to call it products, we can call it like that because we could have a patch of amniotic membrane, and then we have been using as such, and this has been using since a long time, actually, in different places for example, for burns. And there's been used in ophthalmology without actually knowing the mechanism. But the mechanism, I think, is exactly this one. That is this membrane where you have these mesenchymal stromal cells is soak of all these factors that these cells are releasing. So you can either use it as a patch, or you can separate the two populations, the amniotic epithelial cells or the amniotic mesenchymal cells. We are working on this since more than 10 years now, and so we've been setting up all these protocols even with GMP reagents. And I will show you that we are not only working on cells, but on the conditioned medium, so the molecules that these cells are releasing. So I would say these are all the derivatives that you can have and the derivatives that you can use. We have been using so far in preclinical experimentation. Here are some papers mentioned. If somebody of you is interested, I'm very happy to send it or to even discuss new data. So the models that we have been studying is the lung fibrosis, is the myocardial ischemia, the liver fibrosis, and in a recent collaboration with a group in Spain, we have been also studying other models like arthritis, colitis, and encephalomyelitis. And in all these models, we have been applying either cells or conditioned medium or patches, and sometimes both. We have models in which we have tried to use both to see if there was any difference. Of course, I cannot go through all of them, but the general consideration that we had using all this model, and the reason why we went from the patch to the membrane, and from the membrane to the soluble factors that to the factors that are released from these cells is the following. In all these models, what we have observed is that it does have an antifibrotic effect. This is why in the first cartoon where I was showing this progression from inflammation to either a chronic inflammation or to fibrosis, because we do not see the beginning of the fibrotic process. I would say they have an antifibrotic, not because they reduce fibrosis, but because they prevent fibrosis formation. So this was demonstrated by us, but then many other papers came out in the different models that I mentioned to you. Another thing, it was the cell engraftment. It was not really appreciated, mainly when we have used the patch of the amniotic membrane. We did not see any cell migrating from the patch to the tissue where we put our patch. So we thought, well, are now the factors released by these cells the one that play really the game here? Well, when you have an idea, then what you can do, you can just prove it. So we have done the same experiment that we are doing a few years before using cells in this fibrotic, in the mice in which we induce lung fibrosis. But this time we apply, so we have this animal model where with fibrosis, it looks like a rabbit, and this more than a lung. I don't know what it looks like, but believe it, I wanted to say that this is a mice in which I induce with bleomycin a fibrosis. Then we have a group of animal that is treated with control media, so media where it was not with any type of cells. Actually, recently, and we have published, we have been using also other type of cells. We have collected media from other sources, and then we have bleomycin plus conditioned medium of the amniotic membrane. And what have we observed? Studying different parameters that are fibrosis distribution, fibroblast proliferation, collagen deposition, and alveolar obliteration. So if you go to the paper, you will find, here I put the qualitative data, but here I put the quantitative data for you. So the first bar here for the different parameters that I just told you, so the dark bar indicates the mean of the results that we have obtained and the standard deviation in our group of animals. Then in the one the animal treated only with bleomycin, the bar that is coming immediately after, unfortunately this pointer are leaving me alone, it's the mice treated with bleomycin plus a control media, so not the conditioned medium. And there is never a statistic difference, actually not difference at all between these two groups, but you always have a reduction of, instead, for all the other parameters that are different parameters to look at inflammation, like the alveolar obliteration or fibrosis, the one that are collagen deposition, fibroblast proliferation or distribution, you see you always have a decrease. And if you look during the time, you have in the group that is treated only with bleomycin, you have an increase of these fibrosis, as well as in the group treated with control media. But in the group that is treated with amniotic membrane condition, amniotic cells condition medium, you do not have actually the starting of this fibrosis. So I would say this was and this data, so this was a paper that was published 6 years after our first paper, in which we were showing the effect of the cells, but then having evidences in other type of models and from other groups that we did have a very low engraftment. We wanted to see if it was associated to the condition medium. So this is a clear proof that the condition medium per se, here we did not transplant cells, is having this effect. Of course, we can discuss and say this is only in a time window. Maybe a cell, if you leave it, can stay longer, and this is all well taken, and I think maybe therapy is not the condition medium. It could be, it depends. It depends of the model, it depends of the disease, but this was a proof that actually this is playing a part in this anti-inflammatory process. I want to give, Tila will have time, some data about this in vitro study that we have been doing. So from time to time you see change the people and change the picture, because I want to acknowledge the people of my lab, and so this is a way for me to do it, is to show their face and their name on the slide. So what Marta did, I mean, she was showing, here we have a qualitative data and here in a quantitative way. These are data that are published already since many years, that these amniotic-derived cells are able to downregulate a proliferation induced either via T-cell receptor or in an allogenic stimulation. So if you have PBNC, you stimulate with CD3, CD28, you have a proliferation, but if you add amniotic cells, you can appreciate here how much is the difference that is going on. So you have a downregulation of proliferation, and the same is if you have an allostimulation. And this it happened both when cells were put in a contact or in a transwell. Of course, the reason why we were using the condition medium was coming because we did not see cell engraftment, but also because somebody else in the lab, not the one doing the animal model, but the other one, was showing this data, showing that for having this reduction of proliferation of these immunomodulatory properties, you did not need absolutely the contact, was enough to have a separation through transwell. And we have been showing, one could say, did you induce, thank you, did you induce energy, tolerance, what's happening doing that? Well, it's a transient. And this, I think, is good, because we don't want to have some immune system that is blocked. Okay, so you see this is our mixed lymphocyte reaction. I'm sorry for this shift. But when you add amnion, you have a downregulation of this proliferation. But once you remove these cells, these responders are able to respond again toward the party that no, or a third party. Well, Daniele in the lab, what he said, well, the cells do the effect also in a transwell, but in the transwell, they still can receive a message from the cell that are underneath in the well, okay? And he said, if instead we just culture our cells without addition of anything, and we collect it, so what we have been then calling condition medium, the one that we have used in the animal, without addition of anything. What does it happen? It demonstrated that there was a downregulation of this proliferation as well. And the same it was using just the membrane. So here the other message is the cells, not only in contact, not only in transwell, but culture, out of any inflammatory condition. Once you take the condition medium, they do that. And people that are in the audience that are working with mesenchymal cells from bone marrow will say, that's not true. And it's true. I mean, with mesenchymal cells from bone marrow, it's not true, because if you have again your stimulated PBNC, you add amniotic cells or you add, bone marrow mesenchymal cells, you see there is a downregulation of both. If you take conditioned medium of the bone marrow, you do not have downregulation. To have a downregulation of the conditioned medium produced by the bone marrow mesenchymal cells, you need to put it into an inflammatory condition. You need to give interferon gamma. You need to challenge the cells to produce some of these immunomodulatory factors. But you don't have to do this if you use mesenchymal cells from amniotic membrane. So here starts the difference coming. I don't think we needed to win a game. I'm not saying that, but I'm thinking that we are learning that the effect is specific, and the mesenchymal cells, according to where they are coming from, we are all human beings. We are all with the same dignity, but we have different cultures. And I think this is relevant. This is what is beautiful. And I think also mesenchymal cells, they are coming. They receive different messages if they are in placenta or if they are in the bone marrow. And I think that in placenta they get the challenge of the immune system by the fetal maternal tolerance to which they probably contribute, or at least they smell it as a mechanism. So I know, is it really, because I had 25 minutes as a keynote, but if it's over, I will stop. Here I had some slides where I wanted to show that we have been working on data very recently published on understanding the different type of subsets that are involved. So we have been demonstrating that we have a downregulation of the Th1 response. There is actually not much changes in the Th2 response. We have a clear downregulation of Th17, and we have an upregulation of Treg. So this goes just to make people that would be more interested in the mechanisms that are going on. And we have been also demonstrating the fact that these cells not only have on T cells, but also on the monocytic differentiation. And I mean, to make it very, very quick, if you just concentrate on the markers that are specific of the monocytes, that is the CD14, and the specific of dendritic cells, that is the CD1A, if you culture the monocytes toward the differentiation of dendritic cells, with all a bunch of other markers, but let's focus on these two, you have a downregulation of the CD14 and upregulation of CD1A. But if you culture it with the cells in contact, in transwell, or the conditioned medium, we can go through the rest, but concentrating here, you will see that these cells remain CD14. So there is a blockage in the differentiation from monocytes to dendritic cells. And actually, it's coming out here that you have instead an upregulation toward an M2 phenotype. I know this is getting very confusing unless people know exactly what I'm talking about. But concluding, what I wanted to say at the beginning is this one. So we have been always thinking, and I think it is still one way to look at stem cells as cells that are having an importance in the building up of the tissue and in maintaining the tissue homeostasis, and therefore we have been thinking that cell replacement is a therapy whenever we have an injury. So with either differentiation or even we talk about transdifferentiation, I think that mechanism may still be out there, but is not always the key one in the resolution of at least certain type of diseases. And I think we sometimes forget that when we transplant cells, cells are communicating each other, cells see an environment. So all this talking of this cell type, of course, is going to produce some effects. So we have been focused on this release of this factor. There are many other mechanisms that should be considered in this crosstalk. So to conclude, I think that the paracrine effect and this stem cell mediated crosstalk surely adds a lot of complexity to the game or in the regenerative medicine field. But I think it is also adding therapeutic opportunities. And I'm not saying it substitutes cell replacement, but surely it's complementing cell replacement. And with this I finish, and I mean, the people that were involved, I've been acknowledged with their picture on the slides. This is, I would say, the one that have been also playing a role in the lab, many collaboration and funding institutes that are not funding too much, but okay, we thank all of them, and I thank you for your attention.
Questions? Can I just say, this is being recorded, so will you please speak clearly and use the microphone?
I don't have deep knowledge in your field, but the transit effect of the immune system, when you said that effectively the immune system is decreased and don't attack, do you have study in vivo how this transit effect stay in vivo? And my problem is that when this transit effect is lost, the immune system will attack again. So what is your point on that?
Yeah, well, I think it's, it depends which disease we are talking about. If we are, for example, having an acute inflammation and I'm able to repair the tissue, and this is, of course, if you have a genetic defect where you keep having the problem, well, this is unless you have a continuous cure with that, you cannot do anything. And I think a continuous cure can come maybe not with the cells, but with the conditioned medium, because that it could be easy to keep injecting. Of course, it has to be proven, okay? But in other situations where you have, let's say, ischemia in your heart or a brain ischemia, we are working also with the stroke. If you manage to repair the tissue, you don't have always a continuous cause of a trauma. So once you have done it, that's it. So, but important is to do it in the right moment. The window, I think, it's relevant.
I'm sorry, we won't be able to take all these questions because of the time. So I'm awfully sorry.
I will be here the entire day, so for the coffee break, I'm available.
Beautiful presentation, and it really shows a lot of different aspects that are associated with mesenchymal stem cells and what they do. Not all models are inflammatory models, so the inflammatory portion does not apply. But I think what the cells can do is they can show their plasticity and changing from a very primitive cell, the mesenchymal stem cells, into a phagocytic type of cell, which can clean up what's going on in there before repair occurs. That's an important function that I don't think that the soluble materials can do by themselves. Probably not as good as the cells can. And the other thing we need to consider is if we say that there is no transformation into the final cell type, certain things like maintaining turgor and maintaining the stability, like mesangial cells need to provide support, then that is kind of lacking because the mesenchymal stem cells have no way to really do that unless they transdifferentiate. But beautiful presentation.
Thank you. I think I can only comment that in the model that we have been studying, inflammation is at the base, and I think in many diseases. And then I think is as well, because the conditioned medium per se, as I showed, can make an M2 phenotype. So the phagocytes that you are thinking your mesenchymal cells are doing maybe are the phagocytes of your tissue. Then all depends of the conditions, of course. So it's a complex, and
I'm sorry,
I'm very happy that there are many hands raised, and I will be very happy to discuss it afterwards. Thank you so much.
Thank you very much.
Thank you.
Now, the next speaker is Mariam Eslami. Is she here? If she isn't, we can have some more questions. Is she here? Mariam Eslami from
Why don't you go through all of this so you see maybe more people are missing?
But she isn't. This is the one. Right, would you like to ask a few more questions? Because you can do that.
Yesterday was very nice. I left some...
Right. Yes.
Ornella, thank you. That was a great presentation. Thank you. So clearly, this is kind of a magic bullet, so to speak. Makes sense in certain contexts, but I'm a little bit surprised in other contexts. And one thing you've demonstrated that mesenchymal stem cells coming from amniotic tissue, which is pretty much a heterogenic environment, as we all know. and therefore it's expected they have different functions. And looks like your mesenchymal stem cells coming from bone marrow, they're not doing the same what the other mesenchymal stem cells are doing. Giving the outcome that you see, first of all, our exosomes are involved in this, and have you tried to characterize some of the soluble factors? Because that's going to be really an important issue to address, so that we know exactly who is doing what. These are soluble factors. Clearly, we want to know what those soluble factors are, so that you can target the indications of interest.
Sure. First of all, let me make clear, I'm not saying that the cells, there are how can I say? Also mesenchymal cells from bone marrow exert the same effect, but they exert the same effect only if they are put in an environment where there is inflammation. So when you transplant cells in an injury, in ischemia, in fibrosis, in these models that we have studied, that if you transplant mesenchymal cells from amniotic membrane or bone marrow, there is no difference in the effect that they do, because they are releasing these factors, and then I come to the question about which factors they could be. They are releasing these factors, so the mesenchymal cells from bone marrow get a message, and an inflammatory message, and they do it. So there is a crosstalk with the environment. With the amniotic membrane, it seems, and we have demonstrated this with the conditioned medium experiment, it seems that the amniotic membrane doesn't need to receive this challenge. I have to be honest. If it receives this challenge, it's stronger, the effect, okay? Now, if I go back to the presentation, you will see that when there were the cells transplanted, it was more than the conditioned medium effect. So what I want to say is that they are doing similar things, but the amniotic membrane-derived mesenchymal cells are more prone to do it compared to the mesenchymal cells of the bone marrow, and my explanation, it's only philosophy so far, this I agree, is due to the fact that the mesenchymal cells in the bone marrow, they are there, quiet, control the environment, but they are not challenged by the immune system. The mesenchymal cells in placenta, they get a big challenge continuously because of this immune crosstalk between the mother and the fetus. This is, okay, so that's just to make it clear. And this is why I think that so you can prepare conditioned medium of the amniotic membrane cells per se. If you want to take conditioned medium from the mesenchymal cells of the bone marrow, you need to put some interferon gamma. If you put some interferon gamma in the culture, the conditioned medium will do the same than the conditioned medium from amniotic membrane, okay? So after having said that, it is only that it seems that one is being already in this inflammatory condition compared to the other, is coming to the factors, to whatever it is. So there is a big deal about this exosome story. We are trying to see if these are in exosome or not. I mean, at the end, I think like this exosome will be only carrier of whatever is inside. So it's, I mean, okay, they are, I have to be honest, the few experiments that we have done, but I didn't want to put it on a slide because are only very preliminary, and we have done too little. It doesn't seem to be in the exosome fraction, but I mean, take it as something that I said and forget it if you like, because I would need to prove it. Trying to find out which type of factors. We have been published a paper a couple of years ago, and I could make the story short saying that we know very little of what is inside. I can mention IDO, I can mention prostaglandin E. What we have been doing, because we didn't come out, and as all the people that are working with mesenchymal cells of bone marrow, I mean, from time to time comes out a new molecules, but I think is, I don't know, is how these molecules are, I'm not so sure, but nobody has been able to reproduce yet a synthetic conditioned medium like the one that these cells produce. What we have been doing, we have been trying to also block different pathways. So for the synthesis of protein, to see is it a protein, we have been doing some freezing and thawing, heating and inactivating to see. I don't think the major component is of protein because or it is a very small peptide, because you can bring it up to 80 degrees and it's still having the same effect. This is just to give you some hints. It seems like we, for sure, blocking the COX pathway, we could revert this this T cell, let's say, proliferation. We could revert partially, but not completely. We have been doing a characterization of these factors, trying to do a fractionation of the fractionating, so more than 100 kilodalton, 150, whatever, and it seems, again, is something very small. So prostaglandin are small, are there, but are not the only one, because blocking that, it is reverting, but not completely. So that's all what I can say.
Thank you.
Maybe it's thyroxine. You, for a moment, I was taking you for serious.
No, I was just thinking the thyroxine was the one of the early talks, right?
Thank you. But
Ornella, are there conditions during pregnancy where these mesenchymal cells are mobilized either in terms of proliferation or migration?
That's a very nice question. We are studying now these different mesenchymal stromal cells capacity in some pathological pregnancy, like in preeclampsia. And the only thing I could say is that this shift of decrease of Th1, Th17 that we observe here doesn't seem to be, the cells seem to have a different immunomodulatory potential in this pathological pregnancy. But also that one are data on which we are working right now. So,
Last question.
Yes, I think that one candidate for a common mechanism is angiogenicity. Angiogenic mechanism. I don't know what you think about that. And maybe a small comment regarding Transwell experiments. Most of them ignore the juxtacrine effect of the cells, which I think has nothing to do with differentiation, but does transmit important information, which is obliterated by this Transwell experiment. Thank you.
About this Transwell experiment, I could say that, we have the same data with cells in contact with the conditioned medium, but I think, okay, it's relevant. To be careful. If we would have observed something only with Transwell, I would say, well, let's watch out because Transwell can be, but we have observed it at different conditions, so we can put a cross eventually on the Transwell, but I think it's very good to say that, to make that observation. And about the angiogenesis is a very good point, but we haven't done anything about that yet. But thank you.